Oh, you don't do that, or don't do that, because I accidentally go, somebody's like, okay, my pleasure to introduce today's colloquium speaker. I met Brian at the recent hearing loss conference. I didn't realize that articles I had been reading was located just down the street. And when I saw him at the hearing loss conference, I'm like, oh, okay, you should come and give a talk. And I invited him, and then I started looking into his sort of academic past and realized that there were a lot of things that I had read of his in the past that didn't realize were his. I chalk it up to his picture on his webpage because he's clean shaven there and looks like a totally different person. So Brian actually started at the University of Western Ontario, where he, well, I guess he didn't end up there because now it's called Western University Canada.
Yeah, I still got it.
So he did his undergraduate at the University of Western Ontario, and was then working on sort of muscular physiology, neuromuscular junction, and then moved to Virginia Commonwealth, where he worked with Alex Meredith, who I knew very well at the time, and published a really influential paper in my field showing that visual neurons actually, so they don't respond to auditory sounds, but their visual response is modulated by auditory sounds. And that was mind-blowing at the time because you had multisensory stimuli that didn't respond to one of the senses. Subsequently, he went to the University of Buffalo and worked with Dick Salvi, and that's where he got into has been there ever since. He's an associate professor, and welcome, Brian.
Thanks.
So I'm going to use my, as I say, my parental voice. Hopefully it's reception, and we use rodent models for this, and I'll show you how we do that. Students ask me, why did I start studying hearing loss? I used to study the aging neuromuscular system, muscle fatigue. Why did I get into hearing loss? It's prevalence. It's not going anywhere, right? It's going to be something I can study for the rest of my career, hopefully do something to make a positive contribution. Almost 500 million people worldwide have a hearing loss. And what's staggering is the number of children that have a hearing loss. And this isn't just worldwide, this hits close to home in our backyard. But 1 out of every 4 Canadians report having a hearing loss of some type, and it's only going to get worse. These are like the dementia statistics, if you follow folks who do Alzheimer's-related dementia. By 2050, almost 1 billion people are expected or estimated to have a hearing loss. But it's not just an age-related hearing loss we have to be concerned about and how that changes our experiences in life. Developmental hearing loss. This is congenital or inherited genetic basis of hearing loss. In fact, 1 out of every approximate 1,000 births, a child will have a hearing impairment. And 50% of that time, it's because of a mutation in a gap junctional protein in the sensory epithelium of the ear called connexins. Okay, my lab studies a bit of connexin biology. So it's not just age-related hearing loss. It's not just inherited genetic basis of hearing loss. What we do to ourselves through our entertainment or occupation can also render us with a hearing impairment. About 1 billion people are suspected of being in environments that have got loud enough noise to render them with a permanent hearing loss. And in fact, 4 out of every 10 Canadians are going to spend some part of their life working in a place that's just too bloody loud. Okay, you can do it to yourself if you're not careful. A misspent youth too close to rock guitar amplifiers. It's a great way to give yourself an early hearing loss. Not recommended to the young folks in the room. So we can appreciate that with a hearing loss, you're going to impair your communication. And this is a really serious concern for your loved ones and for yourself, but also for the government, because you can lose your workplace productivity. Okay, and as a citizen of Canada, it's important that we have everybody able to contribute. My lab, as David said, got interested in additional features of hearing loss, not just the loss of communication and auditory processing, but the comorbid features, such as tinnitus, that phantom ringing you get in your ears. It's probably more prevalent than you think. About 1% of the population experiences debilitating tinnitus. This is one that can render them without the ability to continue a normal lifestyle. And more research is coming out about the links between hearing loss and dementia. And in fact, an epidemiological study that came out of Johns Hopkins suggested that if you have a hearing loss, it's only about 25 decibels, a shift in your threshold. So modest hearing threshold shift can render your brain 7 years older than it is chronologically. So this is devastating as we move into our golden years. Okay, so I endeavored to design and set up a hearing loss and neuroplasticity lab when I got the opportunity to return to University of Western Ontario. And the ongoing projects in my lab focus on a variety of the things I hinted at. We use rodent models, genetically modified rodents, wild-type rodents, to study such things as the connexin-related mutations. We do that with a neat technique where we actually are able to take the rat pups or mouse pups and pull out their sensory epithelium from their cochlea and keep it alive in its normal organization in a dish so we can study the cochlear hair cells and the spiral ganglion neurons. This is the pathway for audition, and we're able to do this in a cell culture model. But most of the work that my lab does uses actual rodent models to study perceptual features, and the perceptual features that go awry when you have a hearing loss. And we do that with a collection of in vivo electrophysiological techniques. So one of the techniques you'll hear me talk about today is single neuron electrophysiology, where we position a sharp electrode near, in the extracellular space, near neurons that are firing. We can record their single action potentials and how they change. There's another technique we use where we take a multi-channel electrode and we insert it throughout the full cortical thickness. And because we can record simultaneously across this thickness in response to sensory stimuli, we get an idea of the input and output from the particular cortical column. So this gives us an understanding of the feed-forward and feedback connections that occur at the level of the cortex. So that's what we would study at the microcircuit level. In an effort to be translational, we want to try to use electrophysiological recordings that are consistent with what we can do in people. So this down here in the bottom right is a picture of electrocorticography. This is an electrode array that lays on top of the dural surface, similar to what might be done in an epilepsy lab, recording from people who have intractable epilepsy. So the animals can wear these electrodes, and that allows us to get longitudinally recordings like EEG. We do these recordings during behavior because we're interested in the oscillatory activity that goes on in the brain while animals are making perceptual judgments, or we're recording from the neuroplasticity that's at that level. So from single neurons all the way up to cortical oscillations. You can't really study perceptual phenomenon without having animals perform a perceptual task. So I'm happy to talk about this at another time, but we actually train animals to tell us whether or not they do or don't have tinnitus. That was no small feat. I think sometimes it would have been easier to get them to teach them how to talk than it would have been to tell us whether or not they're hearing something that's not real. We study learning and memory and executive dysfunction and how that goes awry with a hearing loss. And what I'm going to talk to you today about is how audiovisual processing and perception changes. We're going to do this with a variety of the electrophysiological tools I've just profiled, as well as some perceptual judgment tasks that David's lab had me experience today, in fact, and I did very poorly. But I want to leave you with an understanding or a discussion on the nature and extent of the neuroplasticity that occurs in the visual system, the auditory system, and the audiovisual system following a noise-induced hearing loss. And I want to, given the audience in your department, I want to highlight our efforts to try to train rodents to do perceptual tasks that are akin to what you can get a group of willing undergrads to endeavor in a psychology department, so that we can better understand the neural basis and the mechanisms of audiovisual perception as well as its plasticity. So one of the things you need to understand when you're studying hearing loss and how it influences auditory and visual processing is that the severity of the hearing loss is very important. Okay, so we're all well aware that the primary auditory cortex in normal hearing conditions processes sound at the single neuron level, at the circuit level, and that ultimately allows you or affords you the ability to appropriately have auditory perception, okay? So a lot of the original work and what happens with hearing loss was done on complete deafness models in either adulthood or congenital deafness. And what that causes to happen is a complete loss of auditory input means that you're left with a deafferented auditory pathway. And what you end up getting is a cross-modal plasticity. Steve Lomber's work, if you're familiar with it, has been very instrumental in helping us understand what's happening when you actually increase the amount of visual responsiveness of that deprived auditory system and auditory pathway. And it can actually provide improved visual processing in the case of motion detection, for example. Okay? What do we see when we have a partial hearing impairment? Well, of course, you still have residual auditory processing. What happens somewhat paradoxically is that the auditory system becomes hyper-excitable. So as long as you're presented sounds that you can hear, a deprived auditory system is actually hyper-responsive to sounds. Okay? We call that central gain. But at the same time, we also see this increased visual responsiveness. So an auditory cortex that has a hyper response to audition and an increased visual responsiveness. Now, if we think about the normal condition, there's areas of the brain that actually are capable of processing audiovisual stimuli. At the single neuron level, what this looks like is neurons capable of reacting and firing action potentials to both auditory and visual stimuli. And these ultimately are believed to be responsible for the audiovisual perception that we experience every day. So my lab wanted to understand what happens when you have a partial hearing loss that occurs in adulthood, very commonly, in our case following noise exposure, at the single neuron level, at the cortical microcircuit level, and ultimately the plasticity as it contributes to audiovisual perception. So if you're going to study audiovisual processing, it's important to understand and appreciate the location of these multisensory neurons. And all mammals have an intersection of their primary sensory cortices where you actually have the multisensory cortex. Okay? It resides and wedged in between the two. In the rodent, there's a lateral view of the rat brain. There's the primary auditory cortex and the primary visual cortex. And wedged in between is the extrastriate visual cortex. This is where the multisensory neurons are to process audiovisual stimuli in the rat brain. So if we take a cross-section of this and then histologically look at it, one of the conveniences of using a rat model is the fact that you can be in multiple cortical regions at the same moment in time, okay? Because of just the lysocephalic brain and the smaller cortical real estate. So what we're able to do is ask the question, at a single neuron level, what's the effect of partial hearing loss on the ability to integrate audiovisual stimuli. And to do that, we use the albino Sprague-Dawley rat, and we use noise exposure to give them a modest hearing loss, a 14 decibel threshold shift. So not a very large hearing loss. And 2 weeks later, we look at what happens to the sensory processing in their visual, multisensory, and auditory cortices. Okay, we use in vivo electrocellular electrophysiological recordings. And these animals are under anesthesia. Okay, that allows us to do these recordings pain-free, but the sensory systems are still fully intact for those of you who don't have experience with anesthetized animal models. So the sensory systems are completely intact. What we do is we're able to insert an electrode, a multi-channel electrode, that covers these 3 cortical regions. I have them in numbers over here on the right. So we can look at what happens into a visual area, progress to a multisensory, typically multisensory area, and finally into a predominantly auditory area, all in the same animal, so we can get a sense of the diversity of the plasticity we might observe. Okay, so how do you study this at the single neuron level? Well, you pick visual stimuli that are effective, a flash of light. It works just as well in us as it does in the rodents. So if you flash a light at a low intensity of 11 lux, or higher, brighter intensity at 82 lux, what we're able to do is record the single action potentials of a group of neurons in response to that stimulus. So here, for the uninitiated, this is a raster plot. So time is along the x-axis. And each one of these rows, from 1 to 50, is the number of trials you'd be presented, in this case, visual stimuli, a bright visual stimulus. And when that visual stimulus is presented, if the neuron happens to be visually responsive, you'll see a clustering of action potentials in the proximity of that visual stimulus, okay? The same thing holds true for auditory stimuli. You can deliver a loud acoustic stimulus at 90 decibels, but because we're dealing with a hearing loss question, we also have to deliver stimuli that's adjusted for the animal's shift in hearing thresholds. It's no good to deliver auditory stimuli to an animal that can't hear it, okay? The neurons don't typically respond to that, okay? So we adjust it. But here's an example where we deliver a loud acoustic stimulus at 90 decibels, and we actually get a reliable response at the single neuron level, okay? So this particular neuron is an exemplar, right? Is a neuron that we might consider bimodal. It responds to 2 modes of stimuli, okay? Audio and visual, okay? But as David mentioned, one of the first studies I did as a postdoc in Alex Meredith's lab was to see whether or not the extent of multisensory processing extended beyond just those overt responses, okay? And you can see that by filling out this matrix, okay? So now we have the bright visual stimulus at the same time as a loud acoustic and every combination in between, okay? And what that allows us to do is find these subtle processing, or find subtle processing of audiovisual information. So here's an example, a classic example of what David was describing during the introduction. Here's a neuron that responds reliably to the visual stimuli. And when you deliver the acoustic stimulus, you get no response. But if you deliver that seemingly ineffective acoustic stimulus at the same time as that effective visual, the visual response is modulated. Okay, in this case, it's suppressed. So that's a form of multisensory processing, and it's quite ubiquitous. Okay, about 25% of the neurons in the multisensory brain respond in this way. Okay, we can see the reciprocal. One that doesn't respond to a visual responds to an acoustic. And in this case, when you take that seemingly ineffective visual and add it to the acoustic, you get a bigger response than you otherwise would. Okay, that's facilitation. And then we have the generic, boring, acoustic-only responsive type neurons. Okay? So by profiling like this, we can see what happens 2 weeks after a modest hearing loss. So to orient you to this slide, what we have are the different neuron categories. Okay, from visual to these integrating ones like suppression or facilitation, these bimodal, they respond overtly to both, and then all the way over to just responsive to auditory. So these are hundreds of neurons from I believe 7 or 8 animals in each group. Okay, and we're able to create this plot of what percentage of neurons are a certain character, a certain type. And no surprise, in gray, in an animal with normal hearing, about 85%, 90% of the neurons in that brain region respond just to visual stimuli. Okay? They might have their visual response modulated slightly, but it's a visual responsive brain region as characterized in this case. And this is from region number 1, this more dorsal area up here in that coronal section. Okay? Nothing happens to that region following hearing loss, okay, in terms of sensory reorganization. But we do see a decrease in the firing rates. Okay? This is something we can talk about afterward. It's quite perplexing. How does a hearing loss cause your responsiveness to visual stimuli in the visual cortex to go down? Okay, it's a very reliable finding. Not quite sure if we understand it. What happens in the multisensory cortex? Well, about 30% of the neurons in the normal multisensory cortex respond to visual stimuli, and about 30% in gray respond to just auditory stimuli. And the remaining third respond to both modalities. Okay, this is true multisensory cortex at the single neuron level. You can see the same thing in ferrets and cats and non-human primates, and presumably in humans. Okay, multisensory cortex. Well, what happens with a partial hearing loss? Okay, we get a dramatic reduction in the number of neurons that are now classified as just auditory. And this isn't because we didn't have the stimulus loud enough. The stimuli are adjusted for the animal's hearing loss. Okay, so turning up the acoustic stimulus still results with a dramatic loss in the number of neurons that are categorized as just being auditory responsive. At the same time, we see dramatic increase in the number of neurons that now solely respond to visual stimuli. Okay, again, these neurons are changing their responsiveness, but not their firing rates. Okay, so what happens now in the normal predominantly auditory cortex, this dorsal auditory zone? Okay, again, even though we've adjusted the acoustic stimulus, we're seeing a reduction in the number of neurons that are responding solely to sound. And what we're seeing is a significant increase in the number of neurons that now respond to both sound overtly and visual stimuli overtly. Okay, an increase in multisensory processing. And what's really interesting is the types of these bimodal neurons show this facilitatory effect. Modest response to visual, a modest response to auditory, and a bigger response to both. And this, we thought, would really matter because now you're combining two modalities of stimuli, and at the single neuron level, in hundreds of neurons, you're getting more firing as a consequence of that. Now, if the language the neurons use to communicate with each other is action potentials, you're getting a lot more action potentials than you used to, okay? But overall, this dorsal auditory zone shows a lot like my earlier work in the primary auditory cortex. You're getting an increased amount of visual processing, okay? The way I think I can leave this slide is that essentially with this modest hearing loss, the multisensory portion of your brain becomes less auditory and much more visual. While at the same time, what used to be predominantly the auditory cortex becomes more multisensory. Okay, and we'll revisit this idea in a few minutes. This increased in visual responsiveness has been coined cross-modal plasticity. Okay? But what happens at the level of the cortical columns, the circuit level, moving beyond one functional unit, beyond the single neurons? So what happens to the cortical microcircuits in a hearing loss in adulthood? This is where we use this technique of recording from the lamina. Okay? We're able to deliver stimuli and record the local field potentials. And then with a bit of math, spatial derivative, what we're able to do is turn that local field potential activity into sources and sinks. And what this does is gives us insight into where electrical activity, current, is flowing into neurons. And this is important because it gives us a sense of what's happening across the different layers of the cortex over time. So here we have time. Okay, here are the stimuli. And we see in red these sinks. This is where depolarization is happening. Current is flowing from the extracellular space into the neurons in the region that is red. Okay? And it's flowing out of the neurons where it's blue. Okay? Sinks and sources. There we go. Okay. What that allows us to do is over time is track the amplitudes of these sinks. And this is important because what it does is gives you insight into the different layers. So here we have the granular sink. This is your classic thalamocortical projection into the sensory cortex, okay? Into the layer 4, right? Up from the thalamus into the sensory cortex. Alternatively, this supragranular sink that's a little bit later, that's intracortical processing. That's the electrical activity caused by intracortical connections, okay? Not the bottom up, but the intracortical activity. So by looking at these peak amplitudes of these sinks over time, we can get an understanding what's happening to the feed forward and feedback aspects following hearing loss. Okay, so we do these recordings Again, one of the advantages of using the rodent model is we can record in multiple fields in the same animal. Okay, and that becomes very important with the paradoxical finding we see at the end. So what do we see in the primary visual cortex? Okay, nothing. The primary visual cortex at the cortical circuit level is not affected, not the supragranular intracortical connections, not the bottom-up thalamocortical inputs. It's unchanged following this hearing loss. But if we move into that more multisensory region in the rat brain, what do we see? Okay, we have the different layers, the supragranular, the granular, and the infragranular. We see what we would call cross-modal plasticity. Now, we're seeing at each one of the layers, the intracortical connections, the thalamocortical recipient layer, we're seeing an increased response to visual stimuli in these circuits following the partial hearing loss. And at the same time, we're seeing a decrease in the responsiveness of these circuits to auditory stimuli. And that's even though we've adjusted the sound, okay, to be relative for both of the groups. So this is an example, this decreased auditory responsiveness and increased visual responsiveness, again, of cross-modal plasticity. We had naturally assumed we'd see the same thing when we moved into this dorsal auditory zone, this area that normally responds predominantly to auditory stimuli in a normal hearing animal. But we see a paradox. At only the level of the granular sink, this is the thalamocortical layer, right? We're getting a paradoxical increase in response. Okay? So if any part of you was considering, well, of course they get a reduced auditory response, the animals have a hearing loss. These hearing loss animals are showing a hyper response to sound in their auditory cortex as a consequence of this hearing loss. That's millimeters away from an area that's actually showing the reciprocal effect. Okay, so the cortical microcircuits is very complex. There's layer specificity as well as differential plasticity. This is an example of that central gain. This is where you're having an increased gain throughout the auditory system following the hearing loss. So the two things occur at the same time, and this is the first study that's actually shown this, right? You can have significant cross-modal plasticity in the multisensory regions of the brain, while central gain enhancement in the auditory regions. And this differential plasticity encouraged us to wonder, what effect is this going to have on perception? Okay, so after a partial hearing loss at the single neuron level, we're getting increased responses to visual stimuli. And at the circuit level, we're getting this paradoxical relationship in neighboring brain regions where there's increased responsiveness to visual stimuli in the multisensory brain region and an increased response to auditory stimuli in the auditory regions. So again, what is this going to do to the animal's level of perception? Okay, so I'm sure I don't need to tell most of you, if you're familiar with the presentations from David and his group, that you can measure audiovisual perception by wondering whether or not stimuli are being bound together as a unified percept. Okay. Again, it's probably not a surprise that if the auditory and visual stimuli occur in somewhat the same space at somewhat the same time, you're more likely to bind them together. As a neurophysiologist, I call that integration. Okay. And experimentally, as we were talking this morning quite a bit about it, you can measure this with judgments on the timing of the stimuli. Okay, a simultaneity judgment, you simply ask the person, did the two stimuli occur at the same moment in time or different moments in time? Or temporal order, a little bit harder to do. I'm abysmal at this. Which one came first? The auditory stimulus or the visual stimulus? So it was clear to us, if we're going to investigate the consequences of cross-modal plasticity and central gain and layer specificity and differential plasticity on these rodents, we have to get them to do what people do. The problem is a task didn't exist to do that. So I had a very talented graduate student, Ashley Shormans. She said, well, I'll just train rats to do what we'll get people to do, okay? And this is how she did it, okay? This is where you actually use an operant conditioning box where a rodent sticks its nose into the center port and waits for the stimuli pair to be delivered, okay? An auditory before a visual or a visual before the auditory. And depending on which stimulus came first, the animal makes an appetitive choice. The left feeder trough, if it thinks that the auditory came first out of the pair, or the right feeder trough, if it thinks the visual came first, okay? And we get the animals to do this, not because they're friendly with us. We get them to do this the same way I get my son, little son, to do things. I bait him with sugar, okay? We give them sugar so that they learn how to do this, appetitive operant conditioning. And this is what it looks like. I hope the sound is appropriate. Okay? So there's a left feeder and a right feeder, and there's the illuminated center nose poke. So the stimuli are going to be 200 milliseconds apart. All right, see if you can do it. So if the visual came before the auditory, the animal goes to the right. The auditory comes before the visual, the animal goes to the left. They are much better at this than I am. So they do this for about 30 minutes a day. We do about 40 animals a day. And they do this like 7-Eleven, 7 days a week. Okay? And very important, we get them so that they're more than 80% correct. Okay? That's when they're at the extremes. Okay, 200 milliseconds apart. But here's the important thing when working with rodents in these types of models. You can't train into them the behavior you want to observe from them. Okay? So we want to know what they think about or what they perceive when the stimuli are not at 200, but at 50 milliseconds apart, or at the same moment in time. And we do that, and result is a psychometric function. Okay? So this is the number of animals, just to orient you to a figure like this. Way over here, this is where the visual is 200 milliseconds out front of the auditory. And the animals are over 90% successful at telling you, yes, the visual was first. And they're equally good at the other end of the curve. Only about 10% of the time, when the auditory is 200 milliseconds in front of the visual, do they think that the visual came first. So that is truly equally good performance. Okay? What we're interested in is what happens when the stimuli are actually physically presented at the same moment in time. And very similar to people, about 50% of the time, they pick that the visual came before the auditory, and the auditory came before the visual. Now, no matter what the rodent picks, they're rewarded for it. This is not training into them. This is probing what they perceive. It's very important, that distinction. So we already saw that this point of when the stimuli are together is about 50% of a pick, auditory, visual, visual, auditory coming first. But to truly believe that this is actually testing rodents for audiovisual perception, we wanted to do a second step, and that is what happens when you manipulate the intensity of the acoustic stimuli. Do you actually see the curves shift? And you do. Okay? Curve shifts to the right if you perceive the acoustic stimuli to be louder. And that makes sense. What that means is that when the stimuli are presented at the same moment in time, you're much more likely to say that the auditory came first if the auditory is louder, okay? Just like in people. And the reciprocal is true. If you deliver what would be perceived as a quieter stimulus, you're more likely to think that the visual came first. Okay? So the rodents experience the same thing when you manipulate intensity as happens in humans. So now we're ready to ask what happens to audiovisual perception when you give them a partial hearing loss. Okay? And importantly, will the curve just simply shift to the left? Because that's the way the curve moves when the acoustic stimulus is quieter. So if you have a hearing loss, this acoustic stimulus is quieter by definition. Will it simply pull your curve to the left? And we'll come back to that in a second. Because it's not that easy to just bring rodents right back in and ask them what happens. You have to prove that in otherwise sham conditions, they can remember the task you trained them for 4 months to do. Okay, so we do extensive sham experiments. So 4 days after the sham, the fake noise exposure, you bring animals back and you ask them, when the auditory is 200 milliseconds in front of the visual, how good are you at telling that was actually the case? They're just as good as they were before you gave them the fake noise exposure. And the same is true in the visual modality. Okay, so the animals clearly remember this task. When it's 200 milliseconds differential between the stimuli, they can remember how to do this. So now we're ready to ask what happens with a real noise exposure. And not surprisingly, when you give them 15 or 20 decibel threshold shift, and you bring them back and ask them, was the auditory stimulus first? They tell you no. not to the same degree. They're making mistakes, okay? And they make a bit of mistakes even on the visual first, okay? And again, not surprisingly, this is related to the level of their hearing loss. So an individual animal that has the greatest increase in its hearing threshold, so the most impaired from the noise exposure, does the worst compared to the animals that had only a modest shift in their hearing threshold. So again, this isn't surprising, okay? You have a hearing loss, you're going to have a harder time with that audio-visual when the auditory is first, okay? But no effect on the visual. Okay. But what happens if you keep doing this performance? You keep coming back and we ask the animal in day 5 and day 6 up to day 10, 12, 13, 14, what happens to your performance with a permanent hearing loss to judging auditory being 200 milliseconds before the visual? Okay? When we ask them what happens to the visual, nothing. They stay equally good at it. But in those days after the noise exposure where they're coming back and performing the task, okay, the deficit they showed gets recalibrated and goes away. So this is an animal, group of animals, with a permanent hearing loss who is now just as good 2 weeks later telling you that the auditory stimulus came first than it was before it had a hearing loss. So this is evidence of a rapid recalibration. Okay? The animal is adapting to the fact that its acoustic input, its afferent input from the auditory system, is now diminished. But its perception starts to come back online. But again, a 200 millisecond deficit is, or a 200 millisecond delay is a large delay. Okay, so this is where we officially ask the question, what happens to the psychometric curve? Okay, and in this case, what we do is we bring the animals back and we get that curve after a sham. We wonder whether or not the curve is going to shift. Okay, but it doesn't. So the animals maintain their audiovisual perception of temporal order judgment despite having a permanent hearing loss. We're able to take this one step further and ask just precisely of what offset between the two stimuli is needed for the animals to tell you in their behavioral responses, when did they think they were actually at the same moment in time? And just like in humans, if the visual leads the auditory by about 15 to 20 milliseconds is when we would say they occurred at the same moment in time. And this is preserved with a hearing loss. So maybe that's not surprising to you, because when you get a partial hearing loss, even a temporary one, it's not like all of a sudden you watch TV and think everything's a badly dubbed kung fu movie, right? You're still able to audiovisually appropriately bind, okay? But think about the level of plasticity that's actually occurring at the neuronal level, right? So we're able to see a behavioral plasticity where we maintain this temporal sensitivity, but we were seeing extensive reorganization at the level of single neurons as well as cortical microcircuits with that central gain enhancement and the cross-modal plasticity. So how is the brain able to maintain temporal precision, right, in the presence of this extensive sensory reorganization? So to answer this question, we go back to using that laminar electrophysiology. And here's actually what happens at the cortical microcircuit level when an auditory and a visual stimulus are presented at the same moment in time. Okay? In your multisensory cortex, there actually occur two separate events. First is the auditory. There's the thalamic layer, right? Thalamic input. And that second sync is the visual. So even though the stimuli are presented at the same moment in time, they hit your multisensory cortex with a significant delay. So what we did was we took advantage of the fact of sliding the timing of the audiovisual stimuli. Okay? And what we see in the rodents is consistent with what's been seen in carnivores and non-human primates is the fact that there's a sweet spot. And that sweet spot is when the visual stimulus precedes the auditory by about 30 milliseconds. That's at the neuronal level where you get the greatest degree of integration. Okay? We did these types of recordings in the areas that were showing changes in our earlier studies. So that's the multisensory zone. So what do we see? Okay, we change the timing. Here's where the auditory and visual stimuli are presented at the same moment in time, and this is where the visual is slid across, such that we get that one response when it's 30 milliseconds in front. Okay, so this shows, oops. Okay, so what do we see in noise exposure? Well, we see the one peak there in blue. Okay, the noise exposed animals in this multisensory cortex, still showing this integration at the level of 30 milliseconds. But we lose the temporal sensitivity. You're getting these very big responses regardless of the timing. Okay. Here's the quantification of that. But what it means is basically the multisensory cortex with just a modest hearing loss loses at the neuronal level, loses its temporal sensitivity. It doesn't matter when you deliver the auditory and visual stimuli in relative timing to each other. You get this giant visual response. Okay, that's consistent with that cross-modal plasticity. But it's important. It's a loss of temporal sensitivity at the neuronal level. What do you see in the predominantly auditory area? Okay, it's neighboring region. Well, this neighboring region is an auditory area. It doesn't care in a normal hearing animal. When you deliver the visual stimuli, it doesn't change the response. Okay, the auditory response is consistent regardless of when you deliver the visual stimuli. And that makes sense. It's the auditory cortex. So consistent at multiple stimulus onset asynchronies. What do you see in noise exposure? Okay, even though you've adjusted the hearing level, this is an attenuation of the responses. So it's a dampening. Less auditory responsiveness in the auditory area, even though you've controlled for hearing loss. Okay, so you're losing audiovisual activity and its responsiveness when you combine the two stimuli together, irrespective of timing. Okay, but there's another technique we can use at this neuronal level, and that is asking about response interactions. And what that is, it's a calculation. So you take a stimulus, let's say the most effective stimulus, in this case, let's say the auditory stimulus, and you ask, how big was the response to that auditory stimulus compared to when I manipulate the timing of the visual stimulus in combination, right? So if you get a lot of response interaction, what that would mean is that adding the visual stimulus causes a big increase in the interactions, okay? And that's classically what we see in normal hearing animals, okay? So that's what we see here. If you deliver the auditory and visual stimuli at the same moment in time, it hits the brain at different moments in time, and therefore there's no multisensory interaction. And there's that sweet spot. When you stagger it and have the visual lead by 30 milliseconds, you get this big multisensory effect. And that's been pervasive over all of the species that have been studied using electrophysiological techniques. So that's completely to be expected. In the neighboring region of the hearing animal, again, the auditory region of the brain doesn't care when you slew the timing of the visual. It just gives you the auditory response, and therefore there's no multisensory interaction. That, again, makes sense. Well, what do we see in the noise-exposed animals in their multisensory cortex? We see, instead of this temporal sensitivity, we see this just large visual responsiveness. So we've lost multisensory interactions in the multisensory cortex. But we gain them in the neighboring region. Okay? We've lost temporal sensitivity in what used to be the multisensory cortex, and we've gained it in what used to be the predominantly auditory cortex. This is a form of compensatory plasticity, where an adjustment is made to deal with the situation of the deafferentation. So it's a compensatory plasticity. So we think that the temporal precision that's actually occurring when audiovisual stimuli are delivered together is actually maintained by a shift in the cortical region that's capable of actually doing that function at the microcircuit level. So this is our working hypothesis. We know that you have a visual cortex and an auditory cortex in a normal hearing animal, and wedged in between is the audiovisual cortex. What we think is happening in hearing impairment, even a modest hearing impairment, you have the visual cortex and you have the auditory cortex, and you have audiovisual cortex wedged in between. It's just physically shifted. The neurons capable of doing what would be considered multisensory processing is just simply moved, okay? So this is a map expansion. The visual cortex is actually larger now, 2 weeks after a partial hearing loss, And the auditory cortex is smaller, following the same hearing loss. And the multisensory cortex is the same size, just moved, okay? But two questions quickly emerge from that proposition: a shift in the functional border of the audiovisual cortex. Is this transition mediated solely through intracortical mechanisms? We're recording from higher order multisensory cortex. Is this simply plasticity occurring at the level of the multisensory cortex? Or are there changes at the level of the thalamus? And the second question is, what's the time course? We look 2 weeks after. Does it persist? How soon does it come online? So the first question, what are the thalamocortical contributions to this cross-modal plasticity? To do this, Ashley took advantage of a pharmacological silencing technique. So she's again recording in control animals with this laminar recording what would be the multisensory cortex. And here she's recording in another group of animals. This is another single example of a noise-exposed animal. And what she does then is she puts on a GABA agonist, GABA A agonist, mucimol, and putting that on the surface of the brain, it's absorbed, and that actually silences intracortical connections. So what that does is it removes the ability of intracortical signaling at the level of this multisensory cortex, and we can see what happens. Okay? And the first thing you notice is dramatic reduction of the activity. The multisensory cortex is multisensory largely because of intracortical connections. Okay? That's the first take-home message here. So the silencing is that you're not getting any more visual response once you've taken away the intracortical connections. And this is what we see in noise-exposed animals. Okay? The first thing is look at the amplitude of this visual response compared to the amplitude of the same visual stimulus in this other group. It's much bigger. So you're getting a much bigger visual response following the hearing loss. This is consistent with our earlier work. Okay? But this is the most important piece. Even though you've silenced effectively the intracortical connections, you're still seeing a granular sync. This is one of the advantages of this technique. This technique measures the input into an area, and that granular sync comes from the thalamus. So this is the first evidence of partial hearing impairment causing thalamocortical plasticity. Okay, it's not just the cortex. I believe cortex is king, or at least that's what my postdoc supervisors told me. Okay, but here's plasticity occurring earlier in the pathway. Okay, at the thalamocortical level. So you're getting input coming in. You're getting input coming in to the deprived multisensory cortex that's already changed with increased visual responsiveness from the thalamus. Okay, and this opens up a tremendous amount of new questions about the mechanisms of thalamocortical plasticity following hearing impairment. Okay, so is it just intracortical plasticity, or is there a possibility of thalamic changes following the hearing loss? This increased visual responsiveness that we routinely see has a thalamic component. Okay, that's the answer to the first question. The second question is, what's the time course of this? Right? Is it hours, days, weeks? And asked another way, this is how the field would ask. This is how my field would ask this. Are there already connections that can support this plasticity? They're already there. They're just masked. And with the hearing loss, they become unmasked, and then they reveal themselves quickly. Okay? So to answer that time course question, we need to change our strategy a little bit. We can't just simply record from animals after. We have to record before and watch the prospect of change. And that requires now chronically implanting electrodes. And that's where I broach the subject of these electrocorticography arrays. These are chronically implanted on the surface of the brain of an animal, and therefore allows us to deliver visual stimuli before and after the hearing loss. So how do we deliver the visual stimuli? It's the same thing. We use a light flash, and it's presented at different intensities. Not surprisingly, as you turn up the intensity of the visual stimulus, you get a sooner and larger visual response. Okay, and in particular, we're focusing on these electrodes here, which is the multisensory region of the animal's cortex. So immediately after a noise exposure, minutes after a noise exposure, we're getting a significant increase in the amount of activity in the multisensory cortex from the same visual stimulus. Okay? And not only is it a larger response, its onset has changed. And so this is parametric. You're seeing a greater response to a visual stimulus moments after depriving the animal aspects of its auditory processing. So what I hope I've been able to show you today is that using in vivo electrophysiology and a variety of different approaches, we're able to see at the single neuron level cross-modal plasticity. This is the increase in visual responses in the multisensory cortex and in the auditory cortex. At the microcircuit level, we see large differential effects. This is where we saw the increase in visual activity in the multisensory region of the brain and a paradoxical increase in auditory responses with that central gain across the cortical layers. And even though we see at the single neuron and the microcircuit level this extensive plasticity 2 weeks after hearing loss, the animals still have intact audiovisual perception. And we think that that perception is preserved because the area of the brain tasked with that temporal sensitivity shifts. Okay? It shifts from what used to be the multisensory cortex more into what used to be more of the auditory cortex. Functional expansion of the visual cortex. So our working hypothesis is that functional border is shifting. Okay? Is that shift purely a cortical phenomenon? No. There's thalamocortical plasticity that's occurring in addition, and that's from the silencing experiments. That increased response in the granular sink, even though you've silenced all of the intracortical activity, is evidence that changes are occurring below the level of the cortex to change multisensory processing. Okay? And the time course of this plasticity is rapid. Okay? Simply in the moments after a noise exposure in a hearing loss situation, you're actually seeing an increased response to visual stimuli that you didn't see before. So this is an unmasking. It's too fast to be simply changes at the level of protein expression. Okay, this is an unmasking of inputs that were already there. So with this, I hope I've been able to show you the nature and extent of plasticity that can occur in the auditory, visual, and multisensory cortices at a variety of different levels following hearing loss. And that you would agree with me, it was worthwhile to have a graduate student train rodents to do what I can barely get my son to do, and that is audiovisual perceptual judgments. Okay, I'm happy to answer any questions, and I'd be welcome to talk about what we're going to do in the future now that we're able to record from animals while they're doing this perceptual task. So thank you very much.
Question about the manipulation with noise-induced hearing loss. I'm wondering, is the hearing loss necessary? Suppose you expose animals long-term to suprathreshold white noise. Do you get any of these changes in multisensory neurons?
So if they were simply like
Daily background?
yeah.
So I'm thinking of the work like Martin Pinkowski and Neil Segerman and stuff, where they're routine exposures. Nobody's ever done it. Nobody's ever looked to see whether or not this routine, daily, or incessant would do it. Judging by the changes you see, though, in the auditory system, the primary auditory cortex, I would think we'd also see them at the level of the multisensory cortex. And if they were then silenced or turned off that noise, I think it would actually go back. Right? I think those inputs, those unmasked inputs would strengthen again. Now, that's a great question. Yeah. Yeah, please. Yes. Mm-hmm. So I think we did that just as a proof of concept. And I believe from pilot experiments, I don't have the full data set. Yeah, so the more salient or efficacious the visual stimulus, the more it convinced the animals that it either came first or second. Yeah. Yeah, we were very considerate of whether or not this was a true finding or whether this was just a context of getting animals to do many trials and just generalizing a psychometric function. So that's insightful. Yeah, please, Zach. Yeah.
Yeah, so you mentioned, not very nice talk though.
Thanks.
You mentioned thalamocortical plasticity.
Yeah.
And I just want to push you a little bit. Are you being careful by saying thalamocortical, or do you actually think that the origin is in changes in response properties in the thalamus?
Well,
Or is it possible the thalamus itself isn't changing, somebody's happening in layer 4 or something like that?
so I think I'm careful with that. Maybe not deliberately, but I'm careful with that. Is that possible? Be careful without being deliberate. I want to be careful because this local field potential source and sink is measuring not the action potentials of the neurons in that region. It's measuring the input coming into that region. That's very important, right? So that input coming in is a consequence of what's firing in the thalamus. That's a crucial piece of anybody who does this laminar analysis with these sources and sinks. Even though you're recording in the cortex, this or the particular region, what you're seeing in those red and blue plots, those blob plots, is the input coming in. Okay, so I'm not exactly sure at this point whether or not it's changes in mechanisms at the recipient layer or whether or not it's actually activity changed at the level of thalamus. That's what we'd have to do is go in with single neurons into the thalamus. I've yet to convince anybody to try to hit the thalamus in these models. Yeah, Dita, yeah, please.
So, sort of along the same lines, the Musol experiment. So, with your silenced cortex and the source sync example that you showed.
Yeah.
There was kind of a smearing of temporal precision. Was that common, or was that just in network?
No, it's common. It's common.
So, and then the average by which the visual input, the visual latency is short, is what, something 5 or 10 milliseconds?
Yeah.
So it doesn't quite cover the roughly 30 millisecond difference. Okay. And do you see a substantial increase in variability in visual latencies? Or
no, they're sort of ubiquitously slowed down. That's one of the things that's happening. We've done other pharmacological studies in otherwise controlled normal hearing animals. And by manipulating the level of inhibition in this multisensory brain region, instead of speeding up visual processing, we actually slow it down. It's got us quite baffled. But yeah, things slow down in this area.
in this model, in your rat, you record typical latency of the ring, but a typical first latency response in B1 with A1. What's the difference?
40 milliseconds? Yeah, so I was telling Dave's group about this. I don't have any data to show you right now, but I think this is really worthwhile for that commentary. When you actually pharmacologically silence the multisensory cortex in a behaving animal, right? What ends up happening is you take that multisensory cortex offline. It can't contribute to the percept anymore. And what you end up seeing in that animal is a shift in their perceptual curve that renders them with what you'd get if you simply looked at the timing of the sensory epithelial responses. So because the acoustic information hits your auditory brain faster than the visual, I showed that with the two responses that we see, okay? If you remove the multisensory cortex, the animal will tell you when the two stimuli came at the same moment that the auditory came well before it. It's almost like it takes that recalibrating portion of the brain offline, and now what you're left with is the physics of those stimuli. We're working on that paper right now. Yeah, Paul.
Right, I was just wondering, what did you use for your noise? Was it long duration noise?
Oh, it was 2 hours of a broadband noise. Yep.
And then all of your work is done with quick testing?
On short noise bursts, 50 millisecond noise bursts.
Well, I'm just wondering, because you're using broadband and broadband damage.
Yes.
We tried focusing the damage with force photo.
Oh,
see
yeah.
if you can figure out where some of these circuit changes are
Yeah.
occurring.
So that's exactly what I want to do. As somebody who studies auditory perceptual deficits following hearing loss, you want to have a high frequency hearing loss. But we didn't know what we were going to get. So we chose the broadband trauma So if we were in a portion of the rat brain that had a bit of a tonotopy, that we wouldn't miss the plasticity. So I'd love to do those studies, just so we're clear, where you're getting a preferential lesion in only the high frequencies to see whether or not the multisensory is now less affected because you haven't dampened the majority of the sensory input from the auditory system. That's really, I mean, that's where I'd like to go because I think that could have huge effects when you start delivering acoustic stimuli of a certain frequency with the visual stimuli. You could imagine if you're using broad stimuli, maybe the perception stays, but if you're using a frequency of stimuli that there is a significant trauma, that would be the region that you might have the greatest plasticity. Yeah, that's great too.
Yeah, the other comment I was going to make is, I know your evidence suggests there is changes in the cell, there could be changes in the cell, but there is also cortical parental feedback.
Yeah.
And so it could actually be one part of the loop.
Right. So that's really important. So that's where I can sort of lead in about what we want to do. My NSERC program is focused on the basic neural basis of this percept. So what we have what we're working on now is taking animals and specifically targeting particular pathways using either chemogenetic or optogenetic silencing techniques where we can tease apart the inputs from particular brain regions so that we can silence them during trials of the perception. And in fact, I was telling the group one of the things that we're most interested in doing is actually taking advantage of a technique called cow light. We're starting this up in the lab, where if you shine blue light on a population of neurons while they're actively firing, they have a calcium transient. What that causes those neurons to do is express halorhodopsin into those cell bodies, meaning that at a subsequent time, shining light in will only silence the neurons that were previously active. So by tagging the neurons during this behavior at particular moments in time, we're going to actually turn off that particular population or that particular projection to see the effect on perception. I'm very interested to know the top-down, anterior cingulate, posterior parietal cortex, the decision-making areas on this perceptual modulation. So now that we have the animal model, now we want to twist and break circuits and then see what happens to that when you actually have a hearing impairment condition. So, yes, stop, yeah.
Think that these processes that you're studying are similar to what might occur in humans and the hearing loss they experience in normal healthy aging?
I
So the audio visual judgments they preserve.
Yes, and there's only a handful of studies that have been done, and it's sometimes difficult to compare them because of what portion of the curve or whether they just use a simultaneity judgment task versus the temporal order judgment task. For a good portion of them, my understanding is that you do have a large preservation of this percept with aging. So I'm not sure if what you're getting in the aging is this slow compensation that's occurring over time, because it took us about 2 weeks for that to finally come back online. We have other work in the lab looking at the age-related hearing loss and wondering what's going to change over time in that particular model. So that's a very real consideration. But whether or not this is what you see in humans, I like to think so, because this is another thing that came up in a discussion. One of the things that happens in this temporal order judgment that we know in individuals is that the prior trial influences what you perceive in the subsequent trial, such that if you were delivered the auditory before the visual in a given trial, the next trial you'll be likely to think the auditory came first. So that's a rapid or a trial-by-trial recalibration. We absolutely see that in the rodents that are normal, typically developing rodents. In our autism models, just like in humans, they lack that. And when we manipulate the level of inhibition at the level of the cortex, we can actually get rid of that trial-by-trial recalibration. So we think what we've got is a consistent perceptual task to the human. So I don't know if that's just. Mm-hmm.
Did temporal order judgments measure the whole psychometric function in undergraduates, 75-year-olds, and their
Yeah, so that's the work that I'm yeah, so that would be what I think. So, but I would think that that's because of the compensatory plasticity that's occurring, right? It's just a little bit more sped up with a traumatic noise exposure. Yeah, David.
When you looked at the rapid recalibration, did you look at that in these noise-exposed animals? Like, so you might think
Yes.
they're
Yes.
calibrating over time, but their
Yes.
rapid recalibration
It's preserved as well.
is preserved as well.
It's preserved as well. Yeah. Yeah. We've only seen the failure to show that trial by trial rapid recalibration if we pharmacologically manipulate the level of inhibition in the particular region that's controlling the behavior, or we have a genetic model of a neurodevelopmental disorder where those patients have that same lack of calibration. So it's a gene mutation that recovers. But if you recover the curve, you recover all aspects of the curve. Was there a question? Yeah, please.
Have you considered behavioral changes in rats when the task is discriminating between sounds as opposed to the TOJ task is really just detecting the sound from
Yeah, so have we, let me try to make sure I understand. Have we done other tasks that separate from the visual where we're getting the animals to tell us aspects about discrimination between acoustic stimuli? Oh, no, we haven't done that. We've done acoustics, solely acoustic tasks where the animals are discriminating between things like amplitude modulation, the intensity. Can they tell that in a noisy environment versus a quiet environment? Kind of like the cocktail party effect. We've done all sorts of that sort of stuff, but never the two together. That's something that would be worthwhile considering. Yeah, Gabriel.
I want to make sure. So you show the behavior at behavior level, then you show the recovery performance over time. It's over almost 2 weeks, right? 14 days. But you can see here at your level, it's just at moments ago that you see this shift.
Yep.
So that's one thing. Since you can see this shift is so fast, why you can see this fast, rapid contribution at behavior?
So this is where I ask you if you reviewed my grant. No, just teasing. Just teasing. No, that's a very real thing. I think what happens is the plasticity occurs rapidly, but what the animal's able to do with that in the decision-making portions of the circuits is what matters. I think that's what takes time, which is why this circuit-specific manipulation with the optogenetic or chemogenetic techniques is going to be so important. Because to put it simply, I think the plasticity occurs rapidly and aggressively, but what the animal's able to do with that takes time in an actual learning capacity. So we've actually done the scenario where we've had animals and we've done the noise exposure, and instead of having them come back and do the training, we simply waited 2 weeks and then tested them. And when you wait 2 weeks and test them, the curve hasn't shifted, but the plasticity's there. So I think this is what do you do with the plastic or the functional reorganization, more so than it's changed and now your perception is now intact. Okay, so I mean, there's a lot of things, but that's how I've rationalized that to a reviewer of my grant, right? Yeah, yeah. Please, back.
So you spend a lot of time talking about single neurons. Do you ever look at populations and, for example, if you get more or less correlated?
Haven't. Okay, I haven't done that, and that is because of, I think, the shortcoming we would see in the anesthetized preparation. So when we go and we've started to do the recordings during the actual behavioral task, that is where we're going to be looking at the synchronized activity across the cortical regions. So that's a great question, but that's predicated on the fact that the animals will be chronically implanted and do the behavior while we record the stimuli. So an awake, behaving, attending animal. I'm really, really concerned about drawing too many conclusions in a ketamine anesthetized, dissociative, non-attentional thing. It's fine for the sensory responsiveness and marking the plasticity. I'd be concerned about what that neural code is to make the extensions to the percept. Yeah. Other questions? Thanks.
Thanks, Brian.